Welcome everyone to this session dedicated to TARMD for the estimation of residence time and determination of the unbinding pathways. So I'm Giulia D'Arrigo and I'm a postdoc in the molecular and cellular modeling group of Professor Rebecca Wade-Titz, and I will guide you today through this hands-on session. But before to go straight to that, I will first, this session will be divided in two parts, let's say. So the first part will be me giving you a short presentation with a sort of bit of theoretical background and introduction on the concepts of TARMD, and then we will have our hands-on session. So I will start with a bit of introduction. Let me show this in mode. Okay, perfect. So, we all know that the affinity between a drug and a target is traditionally based on the binding affinity that is measured at the equilibrium, which means that we consider the drug and target concentrations constant. But we also know that equilibrium in vivo systems, as the ones we are, is very rarely approached. And therefore, in such a case, the concentration at which a drug will interact with the target can vary due to the different physiological processes it goes through. And in this case, in such an open non-equilibrium system, then the individual association and dissociation rates must be taken into consideration, because it has also been shown that the affinity that is measured at the equilibrium often poorly correlates with the in vivo efficacy of a drug, and that instead the pharmacological activity often correlates with the residence time, which means for how long a drug will interact with the target. So this is just a scheme that shows the thermodynamics and kinetics of drug-target interactions. So we start from the two partners in their unbound state, then they go through the formation of transition states to the final bound state. But what I want to highlight, particularly in this graph, is the presence of two important constants, which are kinetic constants, K on and K off, which describe the binding and unbinding event, respectively. And in particular, we are going to focus our attention overall this session, along this session, on the K off, which is here highlighted on the right, which can also be described as its reciprocal, the residence time. And again, why the residence time is important, because the higher the residence time, the longer the duration of interaction between a drug and a target, and therefore the longer the pharmacological activity. But here comes the problem, because if you look at the timescales at which these two events occur, you can see that they are relatively large, especially if we consider the K off, we are talking about hours, and in some cases also days. So you can imagine that this actually represents a problem that we unfortunately all have to face, computationally speaking, because if we look at the timescales of the protein motions, you can see that the two events we are interested in, which are here highlighted by this box, are far beyond the timescale that is accessible with conventional molecular dynamics simulation approaches. And such a computational limit was actually the main motivation for developing TARMD, which was developed in our group by a former postdoc, Daria Hock. And TARMD here, in TARMD, tau stands for residence time indeed, and RAMD stands for random acceleration molecular dynamics. So what do we do with this method is that we are able to speed up the unbinding process and therefore observing an unbinding in a very short time scale. So how does it work? So basically, during a conventional molecular dynamics simulation, we are going to apply a small randomly oriented force to the center of mass of the ligand. The orientation of the force is chosen randomly. Therefore, we are going to check regularly if the ligand displacement is less than a threshold, as the one that is shown here in the slide. And if this is the case, then the orientation of the force will be changed again randomly. And again and again, and then the simulation will go on until the molecule, the drug, has left the binding site. And why this is important, because by looking at the time that our drug is taking to leave the binding site, we are going to derive our residence time. But I'm going to show you this in a couple of slides. Of course, what I want to stress that the magnitude of the force that we are going to choose for a specific ligand must be held constant for all the RAMD replicas that you are going to run for your specific ligand, but also for all the ligands whose residence time needs to be compared. So in the end, what are the advantages of TauRMD? The first, of course, is that we are able to observe a dissociation event in a very short timescale because, as I said just a couple of slides before, typically an unbinding event takes hours or days, and here basically we are speeding up the process, and therefore we are able to see it in the nanosecond timescale. And this is important to stress because, of course, the estimation of the residence time that we get from TauRMD simulation is not an absolute residence time, but is rather a relative residence time because it's, of course, expressed in nanoseconds. Then the second advantage is that we don't make any assumption on the about the ligand egress route because, as I said, the orientation of the force is random. Therefore, we do not bias the direction of the egress. And then only minimum adjustments of parameters are required by the user. So we only advise users to customize the force magnitude according, of course, to their case study. So with TauRMD, we are in a situation where we are then able to simulate the path that goes from the bound to the unbound state. But of course, we want to know what is the actual path that our small molecule is taking while egressing from its protein. And how do we do that? So to do so, we employ the MD-IFP, which is Molecular Dynamics Interaction Fingerprint, which was developed in our group. And what it does, it basically enables to map all the interactions that are established between the drug and the target along the RAMD simulation. So basically, we are going to store all these interactions for every frame and then collect them into a matrix, which will be then converted into a binary matrix as the one that is shown here, where basically 1 means that we have an interaction and 0 means no interaction. And then let's imagine we will have a huge binary matrix for all the frames of all our RAMD trajectories, which will be the input for a clustering analysis. And this is important because through the clustering analysis, we are going to be able to actually see what is the pathway that our molecule is taking. So this is the standard output from clustering analysis. So here you have different nodes which correspond to the different clusters. The size of the cluster, of course, is proportional to the population of the cluster itself. And then you can see that clusters are colored differently. And in this case, this means that the darker the color, the higher is the RMSD of the small molecule with respect to the initial structure. And of course, you might expect that in the unbound state here, okay, in the unbound state here, you have the highest RMSD and therefore the darkest color. Another important thing to observe in this graph is the presence of gray arrows, because these gray arrows indicate the transition between cluster and therefore in the end they outline the total path that our molecule is taking. So for instance here we were able to detect 2 paths, let's say one predominant path, the green one, and a very light one, small one, the Turkish, cyan one. Okay, so anyway, we are going to see this a bit more into detail also during the hands-on session. So here I just want to sort of summarize and try to give you a broad idea of how is our TauRMD protocol altogether. So let's say we want to start TauRMD from scratch. Of course, we need to start from the system preparation that you would do pretty much as you would prepare any system from which you want to start a standard molecular dynamics simulation. And therefore, you are going to minimize it, heat it, and equilibrate it. The second step will be to perform a different standard and deep simulation, because the aim here at this stage is to try to sample as much as we can, try to satisfactorily sample the bound state, because because TauRMD is pretty sensitive to the accuracy of the bound state. So what we do is that we run, let's say, 4, 5 or more standard MD simulation, and then for each of them we are going to get an input structure. This input structure will be used to then run a set of RAMD simulations, as you can see here. So let's say we have 4 input structures that derive from the previous standard MD simulation, and then for each of these 4 structures, we are going to run a set of around 15 RAMD trajectories that we kind of look like the movies that I'm showing in the slide. And this whole set of procedure, basically, of steps will be repeated for every compounds that you want to simulate. So basically, this workflow that I just showed you now is just for one compound. So then once we have collected all the RAMD trajectories, we can perform 2 types of analysis. The first one is to indeed compute or estimate the residence time, the relative residence time. And the way we do it is that we look at the time it takes for half of the RAMD trajectories to be dissociated. So if you look here on the right, you can see in the top plot that we have, let's say, 4 replicas, basically as the one that I showed you in the schema of the protocol on the left. Each replica is colored differently. And basically here, the effective residence time basically corresponds to the mean value where indeed half of the trajectories are dissociated. And then we perform a bootstrapping procedure to finally get our residence time. So this is basically the, in the second plot, you can see the distribution of the residence time for each replica. And from this, we can detect the mean value with the standard deviation, as well as the overall mean for all the replicas, which will in the end correspond to our residence time. And we're going to do this in the hands-on session. Of course, the second type of analysis is the fingerprint analysis and clustering, which will help us in detect what are the pathways that our molecule is taking. And now I will just very briefly show you an application example just to give you a very general and broad idea of what we can actually do with RMD, what conclusion we can get out of it. So the example that I'm going to show you is about the binding of 70 compounds to the N-terminal domain of the histoprotein 19. So here on the left, you can see basically that indeed 70 compounds which have different scaffolds colored differently, and here is basically a plot which shows the range of K-off and K-on values. These are of course experimental values, and you can see that particularly for the K-off, which is what we are interested in because this is what we are actually simulating, you can see that the range is quite wide. In KDBbox, by the way, which is our reference website, you can have also access to an extended data set of compounds and relative experimental data. So here on the right is just the 3D structure of the N-terminal domain of the HSP90 protein. And what I want to show you here is that you can see that the binding site actually can assume different conformations based on what type of ligand is binding. So you can see that for small ligand, we have a more loop conformation of this green helix, and for bigger ligands, we have a more helical conformation. And this you will see now in a couple of slides will actually play an important role for the definition of the residence time. So I will first start showing you by showing you the estimation of the residence time. So this is a plot where we indeed are correlating our computed residence time, which is shown here on the y-axis, and it's expressed in nanosecond because it's relative residence time, against the experimental residence time. You can see that for the 70 compounds altogether, we did get a quite good correlation with an average uncertainty of almost 2.3 of the experimental value. But of course, you can see that there are some outliers, like this green one here. And we actually think that this is due to not sufficient sampling of the bound state. But what we have noticed is that the accuracy of this correlation actually improves when we look at congeneric series, because we said indeed that we have 70 compounds with different scaffolds. Therefore, if we are going to group compounds based on the scaffold, then we do get a higher accuracy and sensitivity of the method. But now let's go to investigate the dissociation mechanisms. So I have just taken here 2 very extreme examples. So compound 8, which has a very short residence time, 5 seconds, and compound 20 with a longer residence time, 3 hours. But again, I want to stress that both 5 seconds and 3 hours are very a very large time that is not so feasible to actually simulate with a standard MD simulation. And what I want to show you here again is basically the same concept of the previous slide. So what we can derive here is that if we have a more helical conformation of the binding site, we might end up with a higher residence time rather than if we have a loop conformation. But now let's have a look at the dissociation trajectory. So if you look at the dissociation trajectory of the fastest compound, you can see indeed that it kind of sort of very immediately, let's say, leave the binding site, while if you look at the compound 20, you can see that kind of gets stuck under this green helix because it's established some interactions. And here you see 2 different trajectories because, as you can see here, our compound is taking 2 different pathways. So it can either egress through the top part or through the bottom part. So now let's go to investigate this a bit more into detail through our clustering analysis. And what you can see here, for instance, for compound 8, you see that it basically immediately leaves the binding site as soon as it indeed loses the main interaction. And in the video that is shown here above, you basically can see the main pathway shown as a blue contour. While for compound 20 on the right, the situation is a bit more complicated. We have more transient states, which corresponds to a high residence time. So let's say we start from the pink cluster here, which is adjacent to the, let's say, bound state. Then from here, we go to the yellow, and then we can egress directly. Or we can either go through the yellow and from the yellow to the blue and then egress, or alternatively, we can also egress through the additional path which go through this orange cluster. And here you can basically see the 2 main dissociation pathway shown as a contour. So the blue one, which goes basically underneath the green helix, and the orange one, which basically goes sort of through the top part. And of course, from these simulations and from this analysis, we can derive some assumptions that at least might hold true for the HSP90 system. So what we have seen is that the steric effects makes the dissociation slower, and that if we have more transient intermediate states between the bound and the unbound state, this might result in a slower dissociation because it means that our compound is spending more time visiting these transient states. So I will now end this first part of the session just with a few take-home messages. So RAMD indeed is an easy and computationally efficient tool for computing relative residence time and also for exploring dissociation mechanisms. And here on the right, you have a plot where we have compared TARMD with other methods that are available in the literature. And basically what this plot is telling you is the amount of time that is needed to simulate the experimental residence time. And you can see that TARMD actually performs quite well. It's among the, let's say, fastest approaches. Only the targeted molecular dynamics is a bit faster, but you should also see that for TARMD, of course, the time that is needed increases as the experimental time, of course, increases. And then I will give you a few hints if you are ever going to use TARMD. The main important notes that I want to give you regards the force magnitude that I said is the only parameter that we advise users to actually manipulate. And we also suggest users to start with a default value of 14 kilocalories per mole that can be then adjusted according to your system. Let's say if the dissociation is too slow, then increase the value, and the other way around. If it's too fast, then decrease it. And then another 2 important points that I want to stress is that multiple replicas is needed because we really need to make sure that we have sampled enough our bound state, because TARMD is sensitive to the quality of the bound state, the accuracy of the bound state. And this is in particular important, let's say, if you start from a docking structure, because in this case, you need to equilibrate more and really make sure that you have sampled enough and equilibrated well. Okay, so this will be basically the end of this sort of introductory part on TARMD. And if you don't have, let's say, any question, I would just move to the, let's say, sort of practical part. So let me know if I can go on. I assume so. Either you write on the chat, but I'm not sure if I can see it, or either you say something. But okay, I guess there are no questions. So I will go on. So the practical session that you are going to perform today, it's about another case study that we have recently published, which is about the GPCRs, which were described quite well by Rui yesterday afternoon during the session. And, you know, GPCR are membrane proteins. And here specifically, we have investigated the behavior of two GPCRs, the beta-2 adrenergic receptor and the muscarinic receptor. In particular, for the beta-2 adrenergic receptor, we have simulated the aprenolol. And for the M2, we have simulated the acetylcholine, the hyperoxo, and the hyperoxo in complex with a positive alloxerin modulator. And what I want to point your attention on is actually the experimental residence time values, because you can see that they span from seconds to minutes. So we are actually simulating quite a long time. And in particular, during the session, we are going to focus more on the muscarinic receptor. And here you can see just a representation of RAMD trajectories that is taken from the M2 receptor with acetylcholine. So now, if we want to start the practical session, I will copy this link in the chat. And it's advisable to have PyMon installed for structure visualization, which you will have during let's say the Jupyter notebook. Otherwise, I can, of course, share my screen and show you the output results if needed. So let me just paste this link in the chat. And please let me know if you are able to see something. You should see something like this. Okay, perfect, thanks. So if we are all on the same page now, so we should be indeed in the first tutorial, which is the tutorial for the computation of the residence time. So what I want to let's say stress is that in this hands-on session we are not going to run any RAMD simulation, because to do it you will need to do it in an external computer or HPC resources. So we are in a situation in which we have, let's say, already computed our RAMD simulation, and we are now post-processing the output from the simulation. The way I would do it is that I would give you a bit of time for you to try it out, and then we will go through it together. One thing that I want to say is that, I mean, it's also a note here, do not run. I mean, if you run the cells all together, then be patient, because there are some visualization cells that might take a while to show. Okay, so if we are all ready, which I guess this is the case, I will give you 15 minutes and then we will go through it together, okay? Okay, perfect. Okay, so thanks. Okay. So of course, the first cell is just to import all the modules that are needed, the packages, and so on. With the second cell, you will create your working directory, which will be here in the main folder. If you click here, then it's in first tutorial, and here you have basically all the materials that you will have downloaded in with the third cells. So Here again, okay, of course, an explanation that I already gave you during the presentation, and the following two cells were just to actually give you a closer view of the system we are dealing with. So this is indeed a muscarinic receptor with in cyan the iprox ligand and in magenta the positive allosteric modulator. And with the second cell, basically, you will see that this is basically the overall system that we have simulated in the end. Of course, between this step here and the next step, basically, there is the overall preparation of molecular dynamic simulation that, as I said, you would do pretty much as you would do for any other cases, and you could do also as Adam has shown in the previous session. So let's say we have run our RAMD simulations, our RAMD trajectories, and we need to post-process the output. So in this case, if we are at point 2, we are at the exact moment where we are going to indeed compute the residence time. So in this work, in this case study, we have investigated the behavior of 3 different force magnitude to 12 kilocalorie per mole, 14 existing, to see how they could affect the residence time. So in these cells, basically, you are asked to say what type of force you want to compute the residence time for, and for what type of ligand. So in this case, we say 2 and for only the muscarinic receptor in complex with the iperoxo. So imagine if we run RAMD simulations, as I said, a RAMD simulation is stopped when the ligand has left the binding site. So this will indeed cause the termination of the simulation, and you will see this termination in the output file that will be written. So if we go, for instance, here in the folder residence time, if we click here and we go, for instance, in the folder we are interested in for the ligand we are computing the residence time for. So here I have just put, let's say, 15 RAMD trajectories starting from the 4th replica. And if we go inside, let's say, this folder, you will see that you have here this out file that I don't know if you managed to open, because it might take a while because it's a very long file, but you will see here at the end you have this sentence here. So this is the sentence we are interested in, because basically what it does is states the number of frames that are executed before the dissociation. And of course, we will have such file for every RAMD trajectories that we run. So for all these 15 trajectories starting from the 4th replicas, as well for replica 1, 2, 3, and 5. So what we do is that we collect this line here in a file that will be a dot file that will look like this. So we have basically a collection of all these lines, and this is the number that we're going to use for the estimation of the residence time. So the way we do it is by executing this Python script, taramd.py, which basically will indeed estimate the residence time. So from the execution of the Python script, you will have an output that will look like this. So here we have 4 different plots. I will try to explain each of them. So in the first plot, you see the cumulative distribution function of the residence time. So as I said during the presentation, we define the residence time as the time at which half of the trajectories are dissociated. And in this case, basically the effective residence time is shown here by this solid red line. But then we perform a bootstrapping procedure, whose results is shown here. So, of course, ideally, if we have sampled enough our system, we would expect a Gaussian-like distribution. And if this is not the case, then we would need to run more replicas and better sample. But we also explored another way of, let's say, estimating the accuracy of the goodness of the sampling, which is basically the Kolmogorov-Smirnov test. And here, in this case, basically, ideally, the KS value should range between 0, 1, and 0, 3. And if this is the case, then we can, let's say, assume that we have performed a good sampling of our replicas. And then after basically we have computed the mean residence time for which replicas, we will be able then to compute the overall mean for all the replicas, which will give us the final residence time, which is here indicated as 20.88 nanoseconds with its standard deviation. So imagine we will do this for every compound we want to investigate, and then we indeed are interested in seeing how good is our correlation with the experimental data. So with this cell, basically you just plot the correlation plot indeed. And here this has been done for the 3 different force magnitude that we wanted to investigate. Here the darker the color, the higher the force magnitude. And you can see that we get a better accuracy with the higher force, but we do not really see too many differences also with lower forces. So anyway, we did get a good correlation. We were quite satisfied with that. Okay, so this is basically the first tutorial that shows you indeed how to compute residence time. So basically, the only thing that you need to do is to collect this out file with the sentence that expresses the time that is taken for the ligand regress, and then collect them into a DAT file that will be then read by the Python script, which will compute the residence time. So now, once we have computed the residence time, we are ready to go ahead. So if you go again into the link that I sent you, which is here, I hope you have it anyway. So in training, Jupyter Notebooks, and if you go into the second tutorial, you should end up in a window like this. So let me know if you're there. By the way, are there any questions for the previous step? Okay, can you tell me if you are in the second tutorial? Okay. Okay, so again, we will do the same as before. So I will give you, let's say 25 minutes, and then we will go through it together. Okay, did anyone else also, has anyone else finished as well? Yes. Okay, then. Okay, so then I will go through it. I hope all of you have finished. Okay. So again, of course, after having loaded all the modules and packages and creating again your own directory and loading the file, so you will be here in theory, the second tutorial, and here. So this is the material you are working on, and we are here. So here, yeah, I have explained a bit better what an IFP is. So again, please pay attention that we do distinguish between, let's say, protein-ligand interaction fingerprints and unspecific contacts, because the HIFP is basically all these specific interactions, let's say hydrophobic contacts, adhesion bonds, and salt bridges, and so on. But we do also compute unspecific protein-ligand contacts, which are all the ones that are found within 5 Armstrong threshold. So as I said during the presentation, for computing the HIFP, we basically create a huge matrix that will collect all the interactions, or indeed all the HIFP and non-specific interaction that are established during the simulation, and in the end we will have a huge matrix. So basically this huge matrix in the end will be in a file, which is a pkl file here, and will kind of look like this. So we will have all the pairwise interactions, as well as all other information like the RMSD for each frame, the center of mass position, and so on. And this PKL metrics will be used for post-processing the analysis, the RAMD simulation. So here, in this case, again, okay, we have defined a different type of interaction, so whole together or only unspecific or specific interactions. And here again, you were asked to select which type of force you wanted to do the analysis for, as well as for the ligand. And we're basically using the same parameter as the one that we chosen before. So here basically we are going to read indeed all the PKL files for each replicas of the same ligand, and what we're going to do in step 2.3 is to check the ligand RMSD and the dissociation from the membrane. So what we're going to do would be to discard all the trajectories where the ligand RMSD is too high at is too small at the very beginning, or if the RMSD of the protein is too high, which means it's very flexible, and therefore we are not going to use those simulations. And we're also going to detect the egress through the membrane. So how do we define egress through the membrane? So we define it if there are like more than 3 contacts with the lipids while egressing in the frame, in the last frames, or if there are less than 2 non-specific contacts with the protein. In this case, for instance, we have discarded 15 trajectories for a pool of 70 trajectories, and we have detected only 1 trajectories where our compound is egressing through the transmembrane helices. So now, of course, we want to actually visualize what are the different pathways that our compound is taking. And to do so, we will first need to manipulate a bit the IFP, because when you start any simulation, specifically with Bromax, which is where RAMD is implemented, the numeration of the sequence will be restarted from 0. Therefore, in this case, what we're doing with the cell is to basically renumbering the sequence, the amino acid sequence, in order to have it basically matching with the crystal structure. So this is easier for comparing and visualization. Okay, and then basically what we're going to do for the clustering, we are going to use the non-specific contact. And the reason for this is that the non-specific contact are, let's say, less sensitive to the relative residue ligand position, and therefore they provide more robust details of the protein ligand contacts during the egressing. So we're going to use those for our clustering analysis. So here it is how we collect all the non-specific contacts into a new matrix, which is XT in this case. And here we're going to perform the clustering. Of course, before to perform the clustering, we need to know what is the optimal number of cluster that is, let's say, suitable for our system. I didn't put it here because it's quite, let's say, long as a calculation, but we have chosen for the system a number of 10 cluster, and we are performing a k-means clustering analysis. So this is where basically you say what type of clustering method you want to use. Of course, you can use different methods and so on. And this is where we actually perform our clustering. So here is where we're going to have the output that I showed you in the presentation. So here you have indeed different nodes. There are differently colored. You have gray arrows. You have, for instance, in particular, 2 main egress routes. This one, which is, let's say, the predominant one, and this other one here. And if you look at this matrix here, this is basically a matrix where all the clusters are plotted against each other. And here, the intensity of the color is basically proportional to the frequency of the transition between clusters. So this means, for instance, that in this case, for this main egress path, we have many frames that actually perform this transition from this cluster here, while we have less frames that perform the transition from this cluster. Then, of course, okay, we have the cluster, but we want to know actually what are the molecular components of this cluster in terms of interactions. And to do so, we compute basically the IFP contacts for every cluster, as well as the non-specific contacts. So with this cell, basically, you have for all the 10 clusters, all the unspecific, the pattern and content of unspecific contacts on the top plot, and of IFP, therefore specific contacts. And then we can save basically these results. I don't know if you managed to open them with PyMol, but I have it here. So I'll try to show them to you. So let's say what we can do in this case is to actually visualize every cluster and its location in the protein. So I'll try to take both windows. So let's say the numbering of the clustering here that you need to look at is here on the y-axis. So, okay, I hope it's not going to be too difficult. So let's say, for instance, here we are in the bound state. We have, let's say, cluster 0. We have cluster 2. So let's show them. So cluster 0, you see, indeed corresponds to the bound state. Then we have, no, cluster 2. We have cluster 1. And then we have also cluster 4. All these clusters basically correspond to the bound state, as you can see here. And they're also indeed colored in a very light color. Then we move, for instance, to the fifth cluster. So we move here. So our ligand is basically moving a bit upward. And then from here, we go, for instance, to cluster 3, which is this one. So this way. And then from cluster 3, we move, for instance, to cluster 9, which is this one, so in this region here, and then from here, basically, it will egress. Or we can also we can also have an egress route through cluster 7, which is shown here. Okay, so basically, you can see that in this way, you can actually have a closer view of what your clustering is referring to within the protein complex. And then, basically, with the next cell, okay, this we already discussed. And then basically, we thought about easier, let's say, way of actually determining the main egress pathway. And the way we did it was to by performing a hierarchical clustering of only the the pre-dissociation frame. So how do we define a pre-dissociation frame? The pre-dissociation frame is defined as the last frame where no more than 3 contacts are, where there are more than 3 contacts. So we collect all of them for all of our RAMD trajectories, and this is basically the pattern of interaction that is found in these pre-dissociation frames, and then we cluster them again through hierarchical clustering. In the end, the outcome that we get is basically looking like this. So basically, we have 2 main egress routes, which is in agreement with what we have seen also in the clustering plot. And again, you have the possibility to visualize them. So I'll show them. Okay. So let's say we have 2 egress routes, so egress 1 and egress 2. And I hope you remember, but so we had cluster 9, which was basically guiding and leading to the main egress path. And then we had cluster 7, the magenta one, which was basically the second populated egress. And if we look at the main egress path, which is this one in red, you can see indeed that goes through the yellow cluster. And if we look at the second egress route, you see indeed that mainly goes through this magenta cluster. So this is basically the end of this second tutorial. If you have any other questions, I don't know, please ask. Or also if everything was clear to you, give me some feedbacks if you want. Did you all manage to visualize the results in PyMon? Or some of you don't have PyMon? Okay, thank you, Nicola. Okay. Okay, thank you. Okay, maybe in the remaining time I can ask you if you have heard about TyrumD before this workshop, or if it was your first time. Okay. Okay, so okay, I see many first time, so it was good to show it. I don't know if there are no further questions. To be honest, I don't know how I should proceed. Björn, maybe I don't know.
Sorry, what was the question?
Apparently, no. Of course, if you have any other doubts, or if you want to try it out by yourself at some point with your system, you can also contact us or me here at the very top of the tutorial. There should be a email. Yes, okay. So if you encounter any problem, or if you're in trouble, you want to start, you can always ask us.